Hi, I'm Fernanda Hashimoto, a medical student at the Universidad Peruana de Ciencias Aplicadas, and I will present a thesis project titled Pharmacophore-Based Virtual Screening Towards the Discovery of Potential Inhibitors of Hepatitis B Virus Capsid Assembly that I've been working on along with my partner Andrea Sánchez. Hepatitis B is a potentially life-threatening liver infection caused by the hepatitis B virus. In 2019, the World Health Organization estimated that 296 million people had a chronic hepatitis B infection, and it resulted in around 820,000 deaths. Chronic infection puts people at high risk of developing possibly fatal complications such as cirrhosis, hepatocellular carcinoma, and liver failure. The drugs available right now on the market are not curative. They have a high rate of resistance, they are costly, and they are limited by their toxicity. Thus, inhibition of the capsid assembly is proposed as a pharmacological target due to its relevance in the virus replication cycle and the lack of approved drugs with this mechanism of action. One of the most studied and interesting families is the heteroaryl hydropyrimidine, or so-called HAPs. We are searching for new hepatitis B virus capsid assembly inhibitors using pharmacophoric models and a massive virtual screening platform. Here we can see the structure of the capsid. We did a bibliographical search and we found 3 crystal structures of the capsid in complex with 3 different HAPs in the Protein Databank archive, with the entry IDs we see here in the screen. And here we can see the general structure of the HAPs. First, we performed a molecular docking protocol validation using Ligand Scout to ensure the accuracy and reliability of our in silico docking experiments. We used Autodock Bina XP with an exhaustiveness of 80, and we then compared the binding configurations of the crystallized ligand in light blue and the docked ligand in purple. The molecular docking protocol regenerated the binding conformation of the capsid ligand complex that was determined by crystallography with an RMSD value of 0.553. We also reviewed interactions the ligand established with amino acids in the binding pocket and compared them with the data available from previous research. The red arrows represent hydrogen bond acceptors, the green arrow represents hydrogen bond donor, and the yellow areas represent hydrophobic interactions. And here we can see how the ligands of the 3 crystals bind to one of the pockets and how they all align. We created 2 pharmacophores, both ligand and structure-based models. We refined them and evaluated if the models could accurately recognize molecules with antiviral activity from those who don't have antiviral activity, using experimentally active and inactive molecules we retrieved during our bibliographical search. We used the area under the curve, ROC curve, to validate our pharmacophores. This graph plots the true positive rate against the false positive rate. The higher the AUC, the better the performance of the model at distinguishing between the active and inactive molecules. Both models had an AUC above 0.9, meaning they have an excellent performance. We then used these models to run a massive virtual screening using the database Chemo28 that contains 2,680,904 molecules that were manually curated, and we used a virtual cluster named Amazon Web Services to make the process faster and more efficient due to the large number of molecules we were screening. The ligand-based model detected 41 hits in less than 3 days, but didn't identify any new candidates. Meanwhile, the structure-based model yielded 459 hits in 3 hours 49 minutes with a hit rate of 0.017%. The 459 hits were analyzed using molecular docking to identify potential hepatitis B capsid assembly inhibitors. We are showing today 2 of the 8 potential inhibitors we have found, the molecules 56 and 73. Again, the crystallized ligand is in light blue, and the purple ones are the docked molecules. Both molecules bind to the pocket with a similar disposition to the crystallized ligand, and they have affinity energies close to that of the HAPs bound to the crystals. We also use the Hashimoto coefficient to compare the similarity between the structure of the hits and the crystallized HAP. Molecule 56 has a coefficient of 0.096, and molecule 73 has a coefficient of 0.15, where a value of 1 represents the highest similarity. So, we want to emphasize that we were able to find potential candidates with different structures than the HAPs we initially used to create the models. And we managed to do so in a rapid and efficient manner thanks to the Amazon Virtual Cluster, whose implementation is a novel approach here in Peru. So, we can conclude that in silico tools such as Pharmacophore-Based virtual screening using a cloud-based setup enable rapid and efficient discovery of potential hepatitis B virus capsid assembly inhibitors. Specifically, 2.6 million molecules were screened, identifying 459 candidates that were analyzed using molecular docking. 8 compounds were selected as potential new candidates and will be further studied by molecular dynamics to determine the stability of the complex ligand protein and used in vitro models.